or share this shit. So lab practical 2 review. First one is the seedless non-vascular plants. What a common name for this group, known as a bryophyte, right? What are they called? Amphibians of plant kingdom. They require water for fertilization. Do they exhibit alternation of generations? Yes, all of the plants do that. What is the dominant plant body? For the seedless non-vascular, that's the gametophyte. The sporophyte literally parasitizing on gametophyte. And then as soon as we start looking at the vascular plants, the roles reverse pretty much. Does the dominant plant body, what we said, gametophyte is a haploid. Gametophytes are always haploid. What are the 2 seedless non-vascular plants you studied in this lab? We talk about Marchantia and we also talk about the moss, right? So here we're looking at the liverworts and mosses. So in the gametophyte, can you identify the gametophyte? That's the whole thing, is the gametophyte right here. You're looking at the gametophyte. Gametophyte will produce those particular gemma cups. Within the gemma cups, you have tiny little bitty part of the thallus, the body of the plant known as the thallus, and it spreads out, covering the area as much as possible. So it's asexual reproduction because the pieces are genetically identical to the plant body. So how does the sexual reproduction take place? Just discuss that. Be able to identify gemma cup right here within the thallus, and the gemmas inside of the slide. Here we go. So here's the cup part, gemma cup, and that's the gemma. Being able to identify anteradial and archegonial head. So let's take a look. Anteradial and archegonial. You see those ones which is kind of sticking out like a palm trees. That's the archegonia. The one which is more like an umbrella is anteradial head. So here we have archegonia. Well, that's archegonia, anteradial. Yeah, again. So here we're looking at the archegonial head. And this is where the sporophytes will start growing on the bottom part. Anteradial head. Here we go, more like an umbrella without those cuts, so to speak. Now we're looking at the anteradial head, and this is the sperm sacs. So inside of those tiny little bitty dots would be a sperm. Now this is what we're looking at, the archegonial head, and this is the actual eggs. The sperm will have to swim, get right there, and this will lead to the formation of the sporophyte. So be able to identify anteradial archegonia. What anteridium bears sperm, archegonia have egg inside. What is the result of the fertilization? Zygote. Where would you find the sporophyte on Marchantia? Since the egg is on the archegonia, on the female plant, the sperm merging, creating zygote that start dividing, so you'll find it on the archegonial head. So, and this is where, this is the cross section of the archegonial head with the 1, 2, 3, 4, and essentially 5 sporophytes growing out of archegonial head. Sporophytes are diploid, and the spores are always haploid, remember that? So this part is the diploid, but spores, myospores, meiosis producing them, those particular tiny little bitty dots are haploid. In the moss, here we're looking, when you're looking at the green part, it's a gametophyte. So all of that gametophyte. Now, from the gametophytes, you will see the capsule coming out. That part is the actual sporophyte, but it's growing out of the female plant again. So we have, that's if it's asking, is it a male or female? Pointing the arrow pointing out. You're looking at the capsule sporophyte grows on the female plant, so it got to be a female. Now, anteridium, so we're looking at the plants, anteridial heads, and this is what we're looking under the microscope. In a microscope, you'll see that anteridial head with the sperm sacs. Tons of those particular sperm sacs, so those dots will be a sperm. Here we go, dot, single dot is the sperm. Water is required for the sperm to swim to get to the egg. So here we go, tons of sperms, because the amount, well, the probability is minuscule, right? Now, in this particular case, we're looking at the archegonial head, and within the archegonial head, you will see this is the single archegonium with the egg inside. That's another archegonium. The egg is not developed yet. This is another archegonium. Here's the egg. That's another archegonium. Here we have the egg as well. And that one in the middle, you can actually start seeing the dark part. That's the zygote. It has been already fertilized. That's the sporophyte start growing. But you don't have to worry about that. As long as you understand the archegonium, the egg, you should be okay. What is absolutely needed for the reproduction? For the reproduction, water is required. The slide of the sporophyte identify capsule and the spore. So here we're looking at the archegonium with the egg inside, and now, so here we're looking at the sporophyte and the capsule of the sporophyte, the top part. So here we're looking at the capsule. So cross section of the capsule. Inside you will see the spores. And that one known as operculum, whenever spores are ready, it will open up, releasing tons of spores. The spores will give rise, because it's a haploid, spores are always haploid, they will give rise to the haploid generation, and you know the haploid generation, nothing else but the gametophyte. All right? Yeah, here we go, tons of spores. Yeah, capsule, seta, spores, operculum. Seta is basically that separation and the thing supporting the capsule. Why it needs to be there? Because again, taking it out higher from the ground, when the spore goes, the wind can easily pick them up. All right, and I think we're done with the lab number 6 overviewing. So lab number 7, seedless vascular plants. So in the vascular plants, what do we see? We're looking at the vascular tissue, the conducting tissue, right? And we were looking at the ferns essentially, and the club mosses. The club mosses will have the stem, underground stem known as a rhizome, will have a tiny little bitty leaves known as the microphyls, and those club-looking structures on the top known as the strobilus. Strobilus will carry spores. So again, same idea, take it away from the ground, so whenever the spores are released, they can be picked up by the air, drop off somewhere, hopefully in a comfortable conditions. So sporophyte, you're looking at the sporophyte overall, strobili at the top part, microphyls a tiny little bitty kind of scale looking leaves. Why they called microphyls in the lycopodium? Because they're small, that's it. Nothing more. Be able to label strobilus, okay, the structure carries what? Spores. Spores are haploid or diploid, always haploid, lycopodium, homosporous or heterosporous. And whenever you're looking at the spores right here, take a look. That, for example, sporangia, and that's sporangia, that's sporangia, and that you can kind of compare. Are the spores about the same size between 3 of those? Yeah, they are about the same dots. Same size dots, homosporous. Nothing else. Homosporous means same size. Heterosporous means the spores will be a different size. That's it. By the way, question on the test. So now, Selaginella. Whenever we're looking at this Selaginella, we're looking at the, again, rhizome, tiny little bitty leaves, almost impossible to see. They are very, very small scale like. But now we're looking at the strobilus on top. So strobilus will have a sporangia with those spores and sporangia with those spores. So I think I have no. Oh, me. So let's kind of take a look on this one. So we have sporangium with tiny little bitty spores, and now we have sporangia with two huge spores. And here's another sporangia with two huge spores. So in this particular case, different size of the spores means that it's a heterospores. We have small spores known as tiny microspores, and they will give a rise to the male gametophyte, and the big ones will give a rise to the female gametophyte. But in this particular case, what main idea is the spores are different sizes, therefore that's a heterosporous plant. Spores are again haploid, and they are heterosporous. All right, let's kind of take a look. What else do we have? So Selaginella showing the microsporangium, megasporangium. Now this guy right here, this is something which is known as a whisk fern. Make sure you understand that. It's a very interesting plant because it resembles, it's not it, but it resembles one of the first plants we ever been able to discover, at least the fossils of the plant known as the Cooksonia. Same kind of idea, stem splitting up and no leaves yet, and on the top it has this sporangia, so yellow dots are sporangium. So psilotum, it should be right here like this. So psilotum, or known as a whisk fern. So sporophyte, the whole thing is a sporophyte, and the yellow part are sporangia with the spores. Equisetum. Equisetum is a horsetail. So here we're looking at the psilotum, aka whisk fern. So horsetails, yeah, I guess kind of look like a horsetail. So here we're looking at the horsetail. Again, horsetails have a very interesting structure of the stem. Stem is hollow, has those nodes, right, where the leaves coming out in the whorled formation. And the very top, it has this sporangia, strobili. Basically a pine cone in the making, kind of. So those strobili taking again the spores away from the ground for the wind to easily pick up. So on the Equisetum, sporophyte, the whole thing is a sporophyte. Strobilus is the top kind of cone-looking structure on top of the plant. What is the arrangement of the leaves Equisetum has? It's more than 3 leaves coming out, or more than 2 leaves coming out from the node, so it's a whorl. Do you see similar arrangement in any other member of this group? Similar arrangements, well, ferns might have it, and also calamites, the ancient horsetails like plants. On the fern herbarium, oops. On the fern herbarium, identify sporophytes. So let's take a look here. We're looking at the strobili and the world of the leaves, right? So on the fern, you're looking right now with the naked eye, that's the sporophyte. Frond, one of those big leaves is the complete frond. So all of that, not one of them, but all of that is the frond. The young frond known as the fiddleheads because they're in the ground, and then they open up and they look like a fiddle violin head, and then they open up, and here we have a frond. Frond, fiddlehead, rhizome. Rhizome is underground stem. And then you will need to find the sorus or sori. So let's take a look. Here we have a bunch of fiddleheads, the fossilized fern showing you the vascular bundles. And now here we have a frond with tiny little bitty dots, right? This is the single one known as a sorus, plural sori. Sorus or sori is a cluster of the sporangia. So locates sporangia. What does this sporangium contain? Spores, of course. So in this particular case here, we're looking at the tons of sporangia. So it's a cluster of the sporangia. Again, cluster, and actually you can start seeing a separate sporangia right there. Again, the same thing. You can clearly see separate sporangia clustered together. And on this one, you perfectly can see. So if it's a cross section, again, you're looking at the tons of different sporangia, and within the sporangia, you will have spores. Spores are, of course, what's the structure within the sporangia? Spores, and spores are haploid. Now, on the Protalus fern, so here we're looking at the single sporangia. On the Protalus, Irish Valentine. Irish Valentine is the gametophyte of the fern, free living. Both of them are free living, not a parasitic. It's green, so it can photosynthesize. Has that particular heart-shaped, tiny little bitty speck of the plant. Has the rhizoids, root-like structures. In the central part, you will identify, well, reproductive structures, archegonia, containing eggs. And on the outer side, those particular dotted look structures, that's the antheridia, containing sperm. So fern Protalus gametophyte, the antheridia, locate the antheridia outside dotted one. What do they contain? Sperm. Archegonia kind of in the middle, they contain egg. Protalus free living. Now, man, it's so messed up. So fossils. Yeah, here we're looking at again antheridia, archegonia, all that stuff. On the fossils, look at this guy. Very, very nice in the lab. It's a big. It's about the size of my hand right there, and it's a horse-like structure, a horse tail-like plant. Used to be about 250 or so millions of years ago. Our previous department head who bought it, she was really happy with it. I'm happy with it as well. It was a really good find, and it's quite often shows up on the lab practical, wink wink. So yeah, be able to identify, again, has the rib lines, has those particular nodes where the whorls of leaves coming out and so on. So again, you can clearly see the stem and the several 1, 2, 3, 4, 5, 6 leaves coming out from a single node, right? So, and you can clearly see the rib lines, and again, we have the stem and several leaves coming out. That's the fern cross section, and here we're looking at the calamite, horse-like plant from about 250 millions of years ago. All right, then we are, I think, finished with number 7. We're going good. Number 8, Gymnosperms. So be able to identify cicada palms or sago palms. Be able to identify male versus female. So here we have a male with the female cone, male cone, and again, in this particular case, it's a dioecious plant, means we have a male plant, well, male plant and a female plant. And here's the male plant. Inside of the male plant, what you will find the pollen. Here we're looking at the pollen grains. Female plant with the female cone, and inside those orange things are actual seeds. So here we're looking at the seeds. So let's see, differentiate between male and female. Cicadas monoecious, dioecious, they dioecious. Seeds. Why are seeds in this group unique? Because now they have a seed, but there is no fruit. Gymnosperms, naked. So they have seed, but no covering of the seed. So naked seeded plants. Now on a ginkgo, identify the herbarium sheet. So if you'll see something like that, again, dog food leaves, that's most likely you're looking at the ginkgo. Why do the municipalities plant only male trees? Because females produce the seeds, and seeds, whenever they ripe, especially falls on the ground, they stink. they stink a lot. They produce a really bad odor. So yes, we are discriminating against females in this particular case for a reason specific. And again, if you're looking at the fossils, once again, if you're looking at that particular, that's the ginkgo leaves, and that one is about 100 or so millions of years old. So look at the fossils. Can you identify, well, the shape as well as the leaf pattern or vein pattern, the fanning one? So here we're looking at the female plant because it has the seeds, right? And this is the female, well, that's actual seed, preserved seed. Now on the pine, here we're looking, identify male cone, female cone. So we're looking at the sequoia, the fossils of the sequoia, and now we're looking at the female cone and the male cone, right? Female, male, heterosporosity, large and small. Now we're looking at the different sporangia, technically speaking, which producing megaspores and microspores. What does male cone bear? What do they produce? They produce pollen. Identify a female cone with seeds. Also, that's the. Why do the pinus have the wings? Pinus leaves, pinus seeds. They have the leaves to fly away from the parent plant. Well, essentially to be distributed by wind, right? So female, male cones. Male cone, and here's inside of the male cone, you will see that tiny little bitty grain of pollen. Female cones with the seeds inside. And here we're looking at the seed. This is the seed itself. And here we have a wing to fly away, glide away, technically speaking. On the slide of male cone, staminate cone, identify microsporangium. So here we go. Let's see. Male cone, cross section. Now we need to microsporangium, a single part right here. That's a microsporangium producing pollen grains, right? Male gametophyte. And within the male gametophyte, you might see the air sacs. It's kind of hard to see because you have to have a huge magnification, so probably will not be on the lab practical, but you can kind of clearly right there see that particular stuff, but you don't have to worry much about that for the lab practical issue. On the slide, let's identify the ovule. So here we're looking at, again, air sacs. Yes, talk about that. So ovule, right? Now, this one is, let me see, here we go. A little bit better picture, stitched with biceral. So the whole thing is the ovule. Now we have a female gametophyte. That's the archegonium, that's the egg. So basically, if you'll know, I don't have, I need to bring, they are from downstairs, the Russian nesting doll, right? The small one, the egg will be located in the archegonium. It will be located in the, well, archegonium will be located within the female gametophyte, all of that located within the ovule. So again, egg, archegonium, female gametophyte, the whole thing is ovule. Yeah. Archegonium, egg, female gametophyte, ovule, and the part which known as the integument. Integument produce the cover for the seed. Not the fruit, the cover. And we're done with number 8. Number 9, really quickly, refreshing your memory, roots will have 3 major parts. Well, 4 major parts, root cap, but 3 major zones: the zone of division, zone of elongation, zone of maturation. Usually include the root hairs, epidermis, cortex. So here we're looking at the vascular bundle, right? Vascular bundle, everything outside is the epidermis, everything between vascular bundle and the epidermis known as the cortex. Pericycle, don't have to worry about that, but that's this one, this particular layer. Vascular cylinder, got that. Pericycle, xylem, phloem. How you can identify xylem? Will have a large openings with a thick walls, and the phloem is right here. Be able to identify different types of the roots. Oops, sorry about that. So here we're looking at the zone of division. That's a root cap. Zone of division right after that. Zone of elongation, and as soon as you start seeing root hairs, that's the zone of maturation. Different types of the root: tap root, fibrous root, and the adventurous root. So here we're looking at the tap root, adventurous roots, and the fibrous roots. That's also adventurous roots. So tap root, adventurous, all kinds of sites, and then the fibrous roots. Root nodules, again, within the root, you will have a bacteria known as the nitrogen fixing bacteria, which help the plants to fix the nitrogen, convert it into form which plants can absorb and build the amino acids, hence proteins. And this is the root mutualistic relationship, quid pro quo. Identify the root nodule. So this is a root nodule, this is the root. What type of the symbiosis between the bacteria we have? That's a mutualism. What does the plant root give to the bacteria? Food and the shelter, sugars and the place to live. Bacteria supply the nitrogen compounds, nitrates. Stems, epidermis, parenchyma. Okay, so epidermis, the outside, then we have a vascular bundle. Within the vascular bundle, you will have a phloem, large vessels, a xylem, and it's surrounded by the sclerenchyma to reinforce the vascular bundle. Pith will be from the, well, in between the vascular bundles. Sclerenchyma 5, phloem, vascular bundle, parenchyma. Parenchyma is basically all of those cells. Cortex outside between the vascular bundles and the epidermis. Pith would be the central part, of course, and the epidermis the outer part. Be able to tell a monocot or dicot. The vascular bundles scattered throughout, that's a monocot right here. That's a monocot again. Vascular bundles are spread throughout. And in the dicots, the vascular bundles will be arranged in a nice ring, clearly separating the pith versus the cortex. So vascular bundles are spread out in the monocots. Vascular bundles are arranged in the ring in the dicots. Vascular cambium is absent in the monocots. Vascular cambium is present in the dicots. That's the reason why it can grow thicker, the lateral meristem, right? Speaking of the apical and lateral meristem, will be questioned on the test. On the woody dicot stem, be able to identify. So here we're looking at the nice arrangement of the vascular bundles. Now, woody stem, that would be the the bark, the outside sapwood is a lighter part, it's a xylem, and then the heartwood is a darker part. On the twigs, be able to identify the nodes. This is where the leaves will attach. There is a node scar, so to speak, or leaf scars. Then the auxiliary buds, there is no terminal bud, and the place between the nodes known as the internode. Leaves, again, simple versus compound. If it's a compound, is it pinnately or palmately compound? You need to know that, right? This is and the venation. Make sure you all understand the venation. Venation and the leaf arrangement is different. So you're looking at the simple leaf with the pinnate venation, simple leaf with the palmate venation, right? Now we're looking at the compound leaf with the pinnate arrangement of the leaflets. We're looking at the compound leaf with the palmate arrangement of the leaflets. Pinnately compound, palmately compound. Don't have to worry. If it's a compound, you don't have to worry about the venation unless it will specifically ask, what is the venation? So read the question. On the models, yeah, and the parallel veins right here, parallel venation mostly for the monocots. Everything else is a dicot. We're looking at this one, that's a pinnately venation and the palmately venation. Palmately simple leaf. So leaf anatomy on the model, make sure you understand that we have a upper lower epidermis, palisade parenchyma, spongy parenchyma, vascular bundle. You're looking, as soon as you'll start seeing palisade parenchyma, you're looking at the dicot leaf. In the monocot, the stromas will be on the bottom in the dicot. In the monocot leaves, they don't have the palisade parenchyma. The stomas will be, or guard cells will be on either side. You have essentially a mesophyll instead of, well, you can call it parenchyma as well. Mesophyll is basically something filling in between the upper and lower epidermis. All right, and the mid vein, of course. And be able to identify if it's a monocot or dicot. Now we're looking at the monocot leaf. Why? Because we have one vein, another vein, another vein, another vein, another vein, another vein, running parallel. So whenever you cross-section, you're seeing this. That's a monocot leaf. Now this is a dicot leaf. Why? Once again, we're looking at the one main vein, central vein, and not much of the smaller veins around. Of course, on a large cross-section, you will see some of those whenever it's palmating, and it will be cut off. But you will have a mid vein and the secondary vascular bundles are much smaller, almost impossible to see. So in this case, you're looking at the dicot versus a monocot. Monocot, tons of vascular bundles. In the dicot, there is only a few. So again, single but large vascular bundle, tons of smaller ones. And we are essentially done. We just talked about the lab number 10. If you want me to, I can go over that really quickly. So flowers, right? Let's see. Flowers, make sure to know the parts of the flower, be able to identify if it's pollinated or not. The sperm, the central part, and the egg knows those parts. Double fertilization, make sure that you know that. Fresh flowers, be able to identify if it's a monocot or dicot. Monocot, dicot, 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 monocot. That one is kind of hard to count the parts, but you can look at the pinnate venation. On the fruits, make sure that you understand if it's a dry or fleshy, if it's a dry, dehiscent or indehiscent, if it's a fleshy or fruity, it goes into a simple fruit, aggregate fruit, and the multiple fruit. Simple fruit is majority, aggregate fruit, strawberries, raspberries, blackberries, and the multiple fruit is the pineapple. On a dehiscent or indehiscent, dehiscent on a dry fruit, dehiscent will open up, releasing the seed, indehiscent state closed. On the part of the seeds, and make sure to be able, I most likely will use the colored one, it's kind of clearly see. So we're looking at the plumule, which will actually lead to the formation of the new leaves, or first true leaves. The hypocotyl, radical, and the whole things are the cotyledons. On the monocot, you have the endosperm, cotyledon, and then we're looking at the embryo with the plumule and the radicle. And we are pretty much done with, well, dispersal, we already talked about it. Large surface area, small seeds, that's most likely wind, some sort of spikes, or flesh, such as berries, that's most likely the animals. And if it has a thick rind like this, it's most likely the water. That's pretty much it for the review. Let me kind of stop recording.